All right, everybody, welcome to the Chapter 10, Section 3 screencast, Regulating the Cell Cycle. So we started out 10.1 with problems associated with cell growth, dealing with the DNA, movement of materials, asexual and sexual reproduction, and then 10.2 we focused on the cell cycle and mitosis. But 10.3, we have to start thinking about how is the cell cycle regulated, okay? Here's what we know. Not all cells move through the cycle at the same rate. When we think about bone cells, nerve cells, skin cells, different types of cells in our body complete the cell cycle, G1, S, G2, mitosis, cytokinesis, at different rates. Now, why is that so? And what actually controls the cell, telling it to divide at certain points and then to stop dividing at other points? Well, early in this study, scientists would grow cells in a laboratory setting. And what they found out is that a cell would continue to divide in a petri dish in a lab setting until it bumped up against its neighboring cell, and then it would stop. So it prompted the question, what is it, internally or externally, that is signaling these dividing cells to stop dividing at a certain point? So for many years, biologists searched for this signal that might actually regulate the timing or the rate of the cell cycle. And in the early 1980s, biologists discovered a protein that was involved in mitosis. And what they did was inject this protein into a cell that was not currently dividing. What would occur in this cell is that spindle fiber or mitotic spindle would begin to form. Now, scientists named this protein a cyclin, and generally understood it as a protein that regulates the timing of the cell cycle. Okay, there's obviously much more to be discovered there, but with that in mind, we could think about some events that could affect the rate of the cell cycle in our body. Here on the right, we have an example of a bone break, which would increase the rate of cells dividing in the area of the injury. Or we can think in terms of development. The idea that we go from a fertilized egg, one cell, to an organism with thousands and thousands of cells, prompting a higher rate moving from young to old. So the discovery of cyclins was just the start. Since then, scientists have identified dozens of other proteins that also help regulate the cell cycle from both inside the cell and outside the cell. With that idea that the cell cycle is regulated from both the inside and the outside, we're going to have our two categories of regulatory proteins. We are going to have internal, inside the cell regulators, and also external, outside the cell regulators. So one group of proteins, internal regulatory proteins, respond to events occurring inside a cell. So what they do is they allow the cell cycle to proceed only when certain events inside the cell have occurred. For example, several regulatory proteins make sure a cell does not actually enter the phases of mitosis until its chromosomes have actually replicated. It's kind of like a timing mechanism. Another regulatory protein, internal, prevents a cell from entering anaphase until these spindle fibers have successfully attached to the centromeres. So there are certain events that internal regulatory proteins look for that allow it to assist in the timing of the cell cycle. Now we also have our external, outside the cell regulators. These are going to be proteins that respond to events that occur outside the cell. Now our most common example of external regulators is known as growth factors. And these are especially important during a couple periods of time: embryonic development and wound healing. What growth factors do is they stimulate the growth and division of cells. So essentially they tell cells to either speed up their cell cycle, or slow down the cell cycle. There are also other external regulatory proteins on the surface of neighboring cells, okay, and they can tell cells when to stop. If a cell begins to butt up against a neighboring cell, our external regulator will indicate, okay, stop the cell cycle, stop dividing at such a fast rate. We are in an area that is highly congested, per se. So they cause cells to slow down or stop their cell cycles. What this does is prevent excess cell growth and keeps body tissues from disrupting one another. Now, when we get to cancer later on down the road, one issue we are going to face is that these external regulators here that tell the cell to stop dividing because of butting up against a neighboring cell, they do not work. So a cell will continue to divide, and we get tumors from that instance. But regulatory proteins, internal and external regulators. Now, just as new cells are produced every day in multicellular organisms, many other cells are going to die. Okay, so we have mitosis and cytokinesis here, which I should note. But many other cells are going to die. So a cell may die by accident due to damage, injury, or a cell may actually be programmed to die. This programmed cell death is what we call apoptosis. Now, once apoptosis is triggered, a cell will undergo a series of steps that essentially leads to self-destruction or death. Okay, so we are going to have the cell and chromatin, that genome or the DNA, will begin to shrink, and then parts of the cell membrane will even begin to degrade. What this does is kind of allows cells to dump its contents out into the adjacent area, and neighboring cells will then quickly clean up the cell's remains. Here on the diagram on the right, you can see the degradation of the membrane, and its contents will soon dump out into the neighboring areas, but they will be cleaned up by those neighboring cells. Now, apoptosis also plays a key role in the development by shaping the structure of tissues and organs in plants and animals. For example, this drawing is going to be a little rough, but you have to hang with me. It plays a key role, apoptosis does, in the formation of the feet of mice. Okay, so consider this an early mouse foot in development. Apoptosis plays a key role because each foot of the mouse is shaped the way it is, partly because cells between the toes, which I will indicate with a highlight, will die via apoptosis during development. So you're going to have certain cells programmed to die within that developing mouse foot to actually give it its shape. So over time, you will have an appendage like a foot that will actually begin to take shape due to that programmed death of the cells in between the toes. Now, when apoptosis does not occur as it should, a number of diseases can actually result. For example, cell loss seen in AIDS and even Parkinson's disease can result if too much programmed cell death, apoptosis, occurs. We've got that new vocab word there, apoptosis. Programmed cell death, series of steps leading to self-destruction. So when thinking about the control of the cell cycle, we also need to think about uncontrolled cell growth, or what happens when the cell cycle is not regulated effectively. Okay, now the principal reason may be that the consequences of uncontrolled cell growth in multicellular organisms can be very severe. And by severe, I mean cancer. So cancer itself is, simply put, a disorder in which body cells lose their ability to control the cell cycle or control cell growth. Now, cancer cells are unable or do not respond to the signals that regulate the growth of most cells, so internal or external regulators. And what happens when these cells are not regulated is you are going to form masses of cells called tumors, which we're all familiar with. We're going to have two types of tumors that we need to keep in mind, and we have probably heard of these terms as well. We are going to have benign tumors, which are tumors that do not spread to surrounding healthy tissue, so you can essentially think of them as pretty localized. We're also going to have malignant tumors, which are considerably more dangerous. Malignant tumors have the ability to invade and destroy surrounding healthy tissue. So another issue with cancer cells, as they continue to spread, they will absorb the nutrients, glucose, water, oxygen needed by other cells. They can block nerve connections and even prevent the organs they invade from functioning properly. So soon, those delicate balances that exist in our body will be disrupted by these masses of cells that exist. So the last piece of the puzzle for cancer is considering what actually causes this uncontrolled cell growth. And simply put, they are defects in the genes that actually regulate cell division. And there are several sources of such defects, which are known as carcinogens, things that can cause cancer, some that we've even heard about from either real-life use or exposure to. A couple examples here. Tobacco and even solar radiation. Now, all cancers have one thing in common. The control over the cell cycle has effectively been broken down. So some cancer cells will no longer respond to external regulators, or some simply fail to produce the internal regulators that the cell relies on to tell it when to divide and when to stop. So defects in genes that regulate cell division lead to not responding to these internal and external regulators. Now, very quickly, we'll touch on some of the current treatments for cancer at this point. The first one here is removal by surgery. Skin cancer, which is the most common form of the disease, can usually be treated in that fashion. Now, melanomas, which you may be familiar with, which is the most serious form of skin cancer, can be removed surgically but needs to be spotted extremely early to do so. Now, other forms of the treatment make use of the fact that cancer cells grow very rapidly and therefore need to copy their DNA quicker than most normal cells. And what this does is make them very susceptible to targeted beams of radiation. And as a result, many tumors can be pretty effectively treated with carefully targeted beams of radiation. And finally, we will look at, or talk briefly about chemotherapy, which is the use of chemical compounds that can kill cancer cells. So for years, medical researchers worked to develop chemical compounds that could kill these cells or at least slow their growth, slow the rate of the cell cycle. And the use of these compounds against cancer is, in fact, known as chemotherapy. And pretty good measurable advances in chemotherapy have taken place in recent years and have even made it possible to cure some form of cancers. But because it targets these rapidly dividing cells, you can also interfere with the function and division of healthy cells that are neighboring that tumor, which is something to consider in this form of treatment. So from this brief stint, we understand that cancer is a very serious disease, and continuing to further our understanding and combating cancer is one of the largest scientific challenges of modern day.